As you can see here, the signals in the hippocampus are struggling to connect. So this is what's causing the MCI.
The clinical minimization. It's just mild forgetfulness. The patient sits in a memory clinic, describing subtle lapses that began over the past year. Appointments occasionally require written reminders. Names take longer to retrieve. A recently read article cannot be summarized with the same precision as before. Daily activities remain intact. Bills are paid. Driving continues safely. Conversation is fluent. The neurological examination shows no focal deficits. Neuropsychological testing reveals scores slightly below age-adjusted norms in delayed recall, but overall independence is preserved. The diagnosis offered is mild cognitive impairment. The reassurance follows quickly. This is not dementia. The distinction is functional. Dementia requires loss of independence. Mild cognitive impairment does not. The brain, however, does not recognize diagnostic categories. It responds to structural and synaptic integrity. By the time measurable decline appears on formal testing, network instability has already begun. Mild cognitive impairment represents a measurable deviation from expected cognitive performance that exceeds normal aging, yet does not interfere significantly with daily function. This definition is clinical. It does not specify underlying pathology. The structural processes driving the decline may already be active for years before formal recognition. Patients often describe an internal awareness that something has changed. They rely more heavily on notes. They repeat stories occasionally. They feel mentally slower during complex tasks. These experiences are subtle and easily attributed to stress or fatigue. The visible preservation of independence reinforces the perception that the condition is minor. However, cognitive performance is the surface output of distributed neural circuits. Memory encoding depends on hippocampal synaptic integrity. Retrieval requires coordinated activity between medial temporal structures and frontal executive systems. Attention relies on intact frontoparietal connectivity. A small reduction in synaptic efficiency across these circuits may produce measurable decline without obvious disability. Neuropsychological testing quantifies this decline statistically. Performance 1 to 1.5 standard deviations below normative means may qualify as mild cognitive impairment. Yet statistical thresholds do not reflect the amount of structural alteration required to produce that shift. Synaptic density may already be reduced. Network synchronization may already be weakened. Reassurance often emphasizes preserved daily function. While independence remains intact, the biological processes underlying decline may continue. Mild cognitive impairment is therefore not defined by severity of pathology, but by absence of overt functional collapse. Imaging at this stage may appear unremarkable or show only subtle hippocampal volume reduction. Standard MRI cannot visualize synaptic loss directly. Functional changes may precede visible atrophy. The absence of dramatic radiological findings contributes to minimization. Some individuals with mild cognitive impairment remain stable for years. Others progress to dementia. The clinical category does not determine trajectory. The underlying pathology does. In many cases, especially amnestic MCI, the structural changes resemble early Alzheimer-type degeneration. The danger of minimization lies in misunderstanding what the diagnosis represents. It does not mean the brain is only slightly affected. It means that the structural damage has not yet crossed the threshold required to impair independence. The first measurable memory decline is not the beginning of degeneration. It is the point at which network inefficiency becomes detectable through testing. The hippocampus may have been under metabolic stress long before this moment. Mild cognitive impairment is therefore a transitional diagnosis. It marks the intersection between compensation and structural erosion. Daily function remains intact because alternative circuits compensate. The underlying pathology, however, may already be established. The reassurance that it is not dementia is clinically accurate. it is not biologically definitive. The brain may already be experiencing pre-dementia network breakdown, while independence remains superficially preserved. Hippocampal network architecture, memory, is a circuit, not a location. Memory is not stored in a single structure. It is encoded, consolidated, and retrieved through a distributed circuit centered on the hippocampus, but extending far beyond it. Understanding mild cognitive impairment requires understanding how this circuit functions before it fails. The hippocampus lies within the medial temporal lobe and consists of interconnected subfields, including the dentate gyrus, CA3, CA1, and subiculum. These regions process incoming sensory information from the entorhinal cortex, which acts as the primary gateway between neocortex and hippocampus. The entorhinal cortex integrates multimodal information and transmits it into hippocampal circuitry for encoding. The dentate gyrus performs pattern separation. It distinguishes similar inputs to prevent memory interference. CA3 supports pattern completion, reconstructing stored information from partial cues. CA1 integrates processed signals and relays output back to the entorhinal cortex, and then to distributed cortical regions for consolidation. Long-term potentiation is the molecular basis of memory encoding within this circuit. When synapses are repeatedly activated, intracellular signaling pathways strengthen synaptic connections, increasing transmission efficiency. This process depends on NMDA, receptor activation, calcium influx, and downstream protein synthesis. Synaptic plasticity allows experiences to be encoded into stable neural traces. The hippocampus does not operate in isolation. It is embedded within the default mode network, a distributed system including medial prefrontal cortex, posterior cingulate cortex, and lateral parietal regions. This network supports autobiographical memory, internal thought, and episodic recall. Coordinated oscillatory activity between these regions is required for successful encoding and retrieval. White matter tracts, such as the fornix and cingulum bundle, connect hippocampus to cortical and subcortical structures. Efficient memory function requires intact structural connectivity. Even subtle degradation in these tracts can reduce communication efficiency. In normal aging, hippocampal volume may decline modestly, but synaptic plasticity remains functional. In mild cognitive impairment, early dysfunction often targets CA1 neurons and entorhinal cortex connections. These regions are particularly vulnerable to metabolic stress and protein aggregation. Importantly, memory impairment in MCI often reflects encoding inefficiency rather than pure retrieval failure. The hippocampus may receive input, but synaptic potentiation is weakened. Memory traces are formed less robustly. Repetition temporarily improves recall because it reinforces weakened circuits. The architecture of the hippocampal network explains why early decline can be subtle. The circuit contains redundancy. Alternative cortical regions can assist with retrieval temporarily. Frontal systems may increase activation to compensate for hippocampal inefficiency. Functional imaging studies show increased prefrontal activation in individuals with mild cognitive impairment during memory tasks. This hyperactivation reflects compensatory recruitment. The hippocampus underperforms. Frontal regions attempt to stabilize output. However, compensation requires energy. Increased metabolic demand may accelerate stress within already vulnerable circuits. The network becomes less efficient and more fragile. The hippocampal system is also time-dependent. Encoding requires rapid synchronization of theta oscillations across hippocampus and cortex. If synaptic timing is disrupted, encoding quality declines even before neurons die. Mild cognitive impairment often represents early destabilization within this architecture. The patient may recall information with cues, but fail spontaneous delayed recall. Recognition may remain intact while free recall declines. These patterns reflect circuit inefficiency rather than total structural collapse. Memory is therefore not a location. It is a network process dependent on synaptic plasticity, structural connectivity, and oscillatory synchronization. When mild cognitive impairment is diagnosed, it often signals that this network is under strain. The decline does not originate in forgetfulness alone. It originates in weakened synaptic potentiation within hippocampal circuits and reduced integration with cortical networks. The measurable drop in test performance reflects early breakdown in a system that once encoded experiences efficiently and automatically. Synaptic failure before neuronal death, the true beginning of MCI. Mild cognitive impairment often begins at the synapse, not with widespread neuronal loss. The earliest measurable dysfunction occurs when synaptic efficiency declines within hippocampal circuits. Neurons may still be structurally present, yet communication between them weakens. In many cases of amnestic MCI, amyloid-beta oligomers accumulate extracellularly in the medial temporal lobe. These oligomers are small soluble protein aggregates that interfere directly with synaptic signaling. They disrupt long-term potentiation, the cellular mechanism required for memory encoding. Even before plaques form, soluble amyloid species impair synaptic plasticity. Tau pathology contributes further instability. Hyperphosphorylated tau detaches from microtubules inside neurons, disrupting axonal transport. When transport slows, essential proteins and mitochondria fail to reach distal synapses efficiently. Synaptic terminals become metabolically compromised. Communication weakens. The reduction in synaptic density can be detected in research settings using advanced imaging or postmortem analysis. Studies show that synapse loss correlates more strongly with cognitive decline than total neuronal count. This means memory impairment may begin before large-scale neuronal death is visible on structural MRI. Functional MRI often reveals altered activation patterns during memory tasks in MCI. The hippocampus may show reduced activation compared to age-matched controls. Alternatively, early in disease, hyperactivation may appear as compensatory recruitment. Both patterns reflect synaptic inefficiency. Cerebrospinal fluid biomarkers frequently show elevated tau and reduced amyloid-beta 42 levels in individuals with MCI who later progress to Alzheimer's disease. These biochemical changes indicate active protein dysregulation, even when cognitive impairment remains mild. Positron emission tomography using amyloid tracers can detect amyloid deposition before dementia onset. Some individuals with MCI demonstrate significant amyloid burden despite preserved daily function. This confirms that structural pathology may precede functional collapse by years. Importantly, synaptic failure is not uniformly distributed. Early vulnerability often affects CA1 pyramidal neurons and entorhinal cortex projections. These areas are central to memory encoding. Damage here reduces the strength of newly formed memory traces while leaving older consolidated memories relatively intact. Because neurons remain alive initially, compensation is possible. Surviving synapses strengthen temporarily. Frontal systems increase activation. Retrieval cues become more effective than spontaneous recall. The patient may perform better when given hints, indicating that encoding was incomplete rather than absent. However, synaptic decline has cumulative consequences. Each weakened synapse reduces network integration. Oscillatory synchronization between hippocampus and cortex becomes less precise. Encoding reliability decreases. Repetition becomes necessary for retention. This stage is biologically active. Microglial activation increases in response to protein aggregates. Inflammatory mediators may exacerbate synaptic stress. Oxidative damage accumulates. The environment becomes progressively less supportive of plasticity. Structural MRI may still appear largely normal during this phase. Hippocampal atrophy may be subtle or absent. The absence of visible shrinkage does not indicate absence of degeneration. Synaptic density reduction does not immediately translate to macroscopic volume loss. The defining characteristic of early MCI is therefore functional inefficiency before irreversible structural collapse. Memory decline at this stage reflects compromised synaptic potentiation rather than total neuron loss. The fear lies in this invisible phase. By the time cognitive testing reveals measurable impairment, synaptic architecture has already begun to deteriorate. The diagnosis of mild cognitive impairment does not mark the start of pathology. It marks the point at which synaptic failure has become detectable through performance. In many neurodegenerative pathways associated with MCI, synaptic loss appears to precede measurable neuronal death. Functional decline precedes atrophy. MCI is often the first clinical sign that hippocampal network integrity is destabilizing at a microscopic level long before widespread structural loss becomes visible. Compensation and cognitive reserve, why performance can appear normal. Mild cognitive impairment often exists in a paradoxical state. Measurable decline is present, yet daily function remains intact. This stability does not mean pathology is minimal. It reflects compensation within distributed neural networks and the protective effect of cognitive reserve. Cognitive reserve refers to the brain's capacity to maintain performance despite structural damage. Education level, occupational complexity, and lifelong cognitive engagement increase synaptic redundancy and network flexibility. Individuals with higher reserve can tolerate greater synaptic loss before functional impairment becomes obvious. When hippocampal synaptic efficiency declines, frontal cortical regions often increase activation during memory tasks. Functional imaging consistently demonstrates hyperactivation in prefrontal areas in early MCI. This represents compensatory recruitment. The brain reallocates processing demand to alternative circuits to preserve output. White matter pathways also adapt. Parallel tracts may temporarily compensate for weakening hippocampal cortical connections. Increased reliance on semantic memory systems can assist with recall when episodic encoding weakens. These adaptations maintain independence despite reduced synaptic plasticity. However, compensation is metabolically expensive. Increased cortical activation requires increased glucose utilization and oxygen consumption. Functional MRI studies show elevated metabolic demand in compensatory regions. This heightened demand may accelerate stress in vulnerable circuits already affected by protein dysregulation. The hippocampus itself may show early hyperactivity before atrophy becomes evident. This hyperactivity is thought to reflect unstable synaptic signaling. Neurons fire more frequently, but less efficiently. Over time, excessive activity may contribute to excitotoxic stress and further degeneration. Compensation masks decline during structured testing. Patients may use rehearsal strategies, mnemonic devices, or slower deliberate processing to achieve adequate scores. Performance may appear acceptable under optimal conditions, but deteriorate under cognitive load. The fragility of this compensation becomes evident during multitasking or stress. When attentional resources are divided, the weakened hippocampal network cannot sustain encoding quality. Retrieval errors increase. Word-finding difficulty becomes more frequent. Slowed processing becomes noticeable. Importantly, compensation does not halt pathology. Amyloid accumulation and tau propagation continue despite preserved function. Synaptic density may continue to decline even as neuropsychological scores remain near threshold values. Cognitive reserve delays threshold crossing, but does not prevent it. Once synaptic loss surpasses the system's buffering capacity, decline accelerates. The transition from mild cognitive impairment to dementia often occurs when compensation collapses. This collapse may appear sudden clinically, but structurally, it reflects gradual exhaustion of network redundancy. When hippocampal volume declines significantly and white matter disconnection progresses, frontal recruitment can no longer sustain performance. The presence of compensation explains variability in progression. Two individuals with similar amyloid burden may exhibit different levels of impairment depending on reserve. However, underlying structural pathology may be comparable. The concept of reserve also explains why reassurance can be misleading. Preserved independence does not equal absence of degeneration. It indicates that compensatory networks are still functional. Mild cognitive impairment represents a state of precarious balance. The brain is adapting to synaptic weakening while pathology advances. Performance appears near normal because additional circuits are working harder to sustain it. The fear embedded in this phase lies in hidden vulnerability. Compensation creates stability on the surface, while structural degradation continues beneath. Once reserve is exhausted, decline becomes visible and often accelerates. MCI is therefore not a static plateau. It is a dynamic stage of compensation under strain. The network is already compromised, but alternative pathways temporarily sustain cognition until structural thresholds are crossed. White matter disconnection is slowing before memory collapse. Mild cognitive impairment is often described as a memory disorder, but the decline rarely remains confined to the hippocampus alone. White matter connectivity plays a central role in sustaining cognitive integration. As synaptic dysfunction develops within medial temporal structures, subtle disruption of white matter tracks further destabilizes network efficiency. The hippocampus communicates with frontal and parietal regions through long association fibers, including the cingulum bundle and fornix. These tracks allow encoded information to be integrated with executive systems responsible for planning, organization, and retrieval. Even minor degradation in these pathways reduces processing speed and retrieval fluency. Diffusion tensor imaging studies demonstrate reduced fractional anisotropy in individuals with MCI, indicating microstructural white matter disruption. This disruption may precede overt atrophy. Axonal integrity weakens before significant volume loss becomes visible. Executive slowing is often the first functional sign of white matter disconnection. Patients may describe difficulty multitasking or organizing complex activities. They can still recall information with effort, but coordination between memory and executive control becomes inefficient. Processing speed declines because conduction velocity across compromised white matter slows. Signals traveling between hippocampus and frontal cortex arrive less synchronously. Memory retrieval becomes delayed not because content is lost, but because integration is weakened. In some cases, small vessel disease contributes to this disconnection. Microvascular changes in deep white matter compound synaptic vulnerability. The combined effect of protein pathology and vascular compromise accelerates network inefficiency. Multi-domain MCI reflects broader network involvement. Attention, language fluency, and executive function decline alongside memory. This pattern often indicates more extensive structural connectivity loss rather than isolated hippocampal dysfunction. White matter disconnection also reduces cognitive flexibility. Switching between tasks requires intact frontostriatal circuits. When these circuits weaken, patients may appear rigid in thinking or slower to adapt to new information. Importantly, white matter degradation is cumulative. As microstructural damage increases, compensation becomes less effective. Frontal recruitment cannot fully offset conduction delays. The system becomes fragile under cognitive load. MRI may reveal mild paraventricular white matter hyperintensities in some individuals with MCI. While often attributed to aging, these lesions represent structural compromise that may interact with degenerative pathology. Memory collapse does not occur abruptly. It is preceded by subtle integration failure. The hippocampus may encode imperfectly, but retrieval inefficiency due to disconnection compounds the deficit. This stage reflects pre-dementia network breakdown rather than isolated memory impairment. The distributed architecture supporting cognition is weakening at both synaptic and structural levels. The patient may still function independently, but tasks require more time. Errors occur more frequently under stress. Conversations require increased concentration. The visible effect is mild. The structural basis is progressive. White matter disconnection transforms synaptic inefficiency into broader cognitive slowing. It marks the expansion of pathology beyond hippocampal microcircuits into large-scale integration networks. Mild cognitive impairment at this stage is not simply a memory complaint. It is evidence that neural connectivity supporting coordinated cognition is thinning. The collapse remains partial, but the trajectory is structural and cumulative. The biomarker phase, when the brain shows disease before symptoms. Mild cognitive impairment often occupies a space between subjective concern and measurable structural pathology. In many individuals, biological markers of neurodegeneration can be detected before cognitive decline becomes severe. This phase demonstrates that network breakdown precedes overt dementia. Amyloid positron emission tomography allows visualization of amyloid-beta deposition in the cortex. Many individuals with amnestic MCI demonstrate elevated amyloid burden, sometimes approaching levels observed in early Alzheimer's disease. This confirms that pathological protein accumulation can be present while daily function remains largely preserved. Cerebrospinal fluid analysis reveals complementary changes. Reduced levels of amyloid-beta 42 suggest deposition within brain tissue. Elevated phosphorylated tau indicates active neuronal injury and cytoskeletal disruption. These biomarker patterns may appear years before dementia criteria are met. Structural MRI may show subtle hippocampal volume reduction in MCI. However, early synaptic loss does not always translate immediately into macroscopic atrophy. Volumetric decline becomes more evident as neuronal death progresses. Functional imaging often reveals altered metabolic activity in medial temporal and posterior cingulate regions. Fluorodiaxygallose PET scans may show reduced glucose utilization in individuals with MCI who later convert to dementia. This hypometabolism reflects reduced synaptic activity rather than complete structural loss. Importantly, biomarker positivity does not guarantee immediate progression. Some individuals with abnormal amyloid PET remain cognitively stable for years. The presence of pathology indicates vulnerability, not immediate collapse. Progression depends on rate of tau propagation, vascular contributions, and individual reserve. The biomarker phase underscores a key principle. Structural disease can precede clinical disability by a significant margin. The absence of functional impairment does not imply absence of degeneration. It indicates that compensatory systems are still operating. Risk stratification in MCI increasingly relies on biomarker profiles. Individuals with positive amyloid and tau markers have an increased likelihood of progression compared with biomarker-negative individuals. Those without these markers may remain stable or reflect non-Alzheimer etiologies. This stage highlights the transitional nature of MCI. It is often the first clinical point at which biological evidence of neurodegeneration becomes measurable. The network has begun to destabilize at molecular and metabolic levels. Early identification of biomarker positivity offers a window for potential intervention. However, therapeutic options remain limited. Disease-modifying treatments targeting amyloid or tau may slow progression modestly, but do not reverse established synaptic loss. The fear associated with this phase lies in its invisibility. The brain may already contain significant protein pathology, while daily independence is intact. The diagnosis of mild cognitive impairment may therefore represent an early warning of structural vulnerability. Biomarkers reveal what cognitive testing alone cannot. They expose the biological processes underlying pre-dementia network breakdown. Amyloid accumulation, tau propagation, and metabolic decline can be active long before functional thresholds are crossed. MCI is thus not simply a clinical observation. In many cases, It is the first measurable manifestation of underlying degenerative pathology that has already begun altering hippocampal and cortical circuits. The biomarker phase demonstrates that degeneration can be present without overt dementia. The brain may be biologically compromised while outward performance remains near normal. This is the transitional zone where compensation and pathology coexist. Understanding this phase clarifies that mild cognitive impairment is not benign forgetfulness. It is often a structural warning sign that the neural network supporting memory and executive integration is already undergoing measurable degeneration. Threshold crossing. When mild cognitive impairment converts to dementia. The transition from mild cognitive impairment to dementia is not defined by a new pathology. It is defined by threshold failure. The biological processes that began years earlier, synaptic loss, tau propagation, white matter disconnection, continue until network capacity can no longer sustain independent function. In early MCI, hippocampal neurons may still be present despite synaptic weakening. Over time, persistent tau pathology spreads transsynaptically from the entorhinal cortex into CA1 and beyond. Neuronal death increases. Structural atrophy becomes measurable on MRI. The hippocampus visibly shrinks. As hippocampal volume declines, encoding failure becomes consistent rather than occasional. Repetition no longer restores recall reliably. Recognition cues become less effective. Memory traces are not consolidated sufficiently to be retrieved later. This marks collapse of encoding integrity. White matter pathways also thin progressively. The fornix and cingulum bundle lose axonal density. Communication between hippocampus and prefrontal cortex weakens further. Executive compensation becomes insufficient to offset retrieval inefficiency. Functional independence begins to erode subtly. Complex tasks such as financial management, medication organization, and navigation become inconsistent. Family members notice increased repetition and confusion in unfamiliar environments. The diagnostic threshold for dementia is crossed when these deficits interfere with daily life. The transition often appears abrupt clinically, but structurally it is cumulative. Years of synaptic decline and neuronal loss finally surpass compensatory reserve. When network integration drops below a critical threshold, clinical performance often deteriorates more noticeably. Metabolic imaging during conversion frequently shows expanded hypometabolism in temporoparietal and posterior cingulate regions. This indicates that degeneration has extended beyond medial temporal structures into broader cortical networks. The loss is not limited to memory circuits. Attention and language may decline as pathology spreads. Executive control weakens further. Behavioral regulation may become impaired. The network fragmentation becomes multi-domain. Importantly, irreversibility becomes evident at this stage. Neuronal death cannot be reversed. Gliosis fills spaces left by degenerating cells. Structural MRI shows cortical thinning and ventricular enlargement. Therapeutic interventions at conversion may slow symptom progression modestly, but do not restore lost synapses. The disease has advanced beyond early synaptic dysfunction into widespread structural alteration. Threshold crossing represents failure of compensation. Cognitive reserve can delay this point, but cannot prevent it once structural loss becomes extensive. The difference between MCI and dementia is functional independence, not pathology onset. This transition clarifies the true nature of mild cognitive impairment. It is not a mild condition in isolation. It is a pre-dementia phase, during which network breakdown has begun, but has not yet disrupted daily life significantly. When conversion occurs, it reveals how long degeneration has been unfolding silently. The patient and family perceive a shift. The biology has been advancing for years. Mild cognitive impairment converts to dementia when hippocampal atrophy, synaptic depletion, and white matter disconnection collectively exceed the brain's buffering capacity. The visible impairment marks the culmination of structural decline that began long before the label changed. The fear lies not in the moment of diagnosis. It lies in the recognition that by the time dementia is declared, the network collapse has already been progressing through irreversible stages for years. Non-memory involvement, attention, language, and executive spread. Although mild cognitive impairment is frequently identified through memory complaints, the breakdown rarely remains confined to hippocampal circuits. As degeneration progresses, additional cognitive domains become involved. This expansion reflects the spread of pathology across distributed cortical and subcortical networks. Attention is often affected early, but subtly. Sustained attention requires coordinated activity between frontal cortex, parietal cortex, and thalamic relay systems. When white matter connectivity weakens and synaptic efficiency declines, attentional stability deteriorates. The patient may appear distractible or mentally fatigued under prolonged cognitive demand. Language fluency may also decline in multi-domain MCI. Word retrieval slows. Verbal output becomes less spontaneous. This reflects dysfunction within temporoparietal language networks and their connections to frontal executive regions. The deficit is not aphasia in the classic sense. Comprehension remains intact, but retrieval becomes inefficient. Executive dysfunction becomes more pronounced as frontostriatal circuits weaken. Planning multi-step activities becomes effortful. Mental flexibility declines. Switching between tasks requires deliberate concentration. These changes signal broader network fragmentation beyond medial temporal structures. The pattern of spread varies by underlying pathology. In Alzheimer-related MCI, degeneration often extends from hippocampus into lateral temporal and parietal cortex. In vascular-related MCI, white matter disconnection may dominate early. In Lewy body-associated MCI, attention and visuospatial deficits may appear prominently. This variability underscores that MCI is not a single disease. It is a clinical stage representing early network instability due to diverse pathologies. The common feature is that synaptic and structural breakdown remain insufficient to impair independence fully. Visuospatial skills may decline as parietal regions become involved. Patients may misjudge distances or struggle with complex visual tasks. These changes are often subtle initially and detectable only through structured testing. Importantly, multi-domain involvement indicates greater structural vulnerability and higher risk of progression. When impairment extends beyond isolated memory circuits, it suggests widespread network compromise. Cortical thickness measurements may reveal early thinning in association areas. Functional connectivity analyses demonstrate reduced coherence within the default mode and frontoparietal networks. These objective findings correlate with subjective complaints of mental slowing. Despite this expansion, many patients remain independent because compensation continues. Frontal recruitment intensifies. Behavioral strategies adapt. However, the network becomes increasingly fragile under stress. Non-memory involvement transforms MCI from a focal hippocampal issue into a systemic pre-dementia state. The brain's integration capacity declines gradually across domains. Memory may remain the most noticeable symptom, but attention, language, and executive coordination are often quietly deteriorating. The presence of multi-domain deficits often signals approaching threshold crossing. Compensation across multiple systems becomes more difficult. Structural redundancy diminishes. Mild cognitive impairment therefore evolves from localized synaptic inefficiency into distributed network fragmentation. The expansion beyond memory circuits reflects spreading structural compromise. The visible complaint may still be forgetfulness. The underlying reality is broader. Attention, language, and executive circuits are increasingly destabilized. The collapse is no longer isolated. It is becoming systemic. This stage reinforces the central concept. MCI represents pre-dementia network breakdown. The damage is not confined to one region. It is progressively affecting the architecture that allows coordinated cognition across domains. Irreversibility. Why the early intervention window is limited. Mild cognitive impairment represents a narrowing window between reversible synaptic stress and irreversible structural loss. Early in the process, synaptic dysfunction may still allow partial recovery if pathological drivers are slowed. However, once neuronal death and tract disconnection become substantial, restoration is no longer biologically feasible. Synaptic density reduction precedes measurable atrophy, but it is not indefinitely reversible. Long-term potentiation depends on intact dendritic spines and stable cytoskeletal support. When tau pathology disrupts microtubules and amyloid oligomers, impair receptor signaling persistently, synaptic architecture degenerates. Dendritic spines retract. Plasticity declines permanently. Neuronal death marks a critical transition. Once hippocampal pyramidal neurons are lost, adult neurogenesis is insufficient to restore the original circuit capacity. Adult neurogenesis within the dentate gyrus is limited and cannot compensate for widespread loss. White matter disconnection compounds this irreversibility. Axonal degeneration within the fornix and cingulum bundle reduces communication between hippocampus and cortex. Gliosis fills areas of structural loss. Central nervous system axons have extremely limited regenerative capacity and do not functionally reconstitute long-distance tracts across established gliotic barriers. Network integration becomes permanently compromised. Disease-modifying therapies targeting amyloid or tau aim to slow progression rather than restore structure. Even when amyloid burden is reduced pharmacologically, pre-existing synaptic loss and neuronal death remain. Removing plaques does not reconstruct destroyed dendritic trees. The concept of threshold applies here as well. Early in MCI, synaptic dysfunction may fluctuate. Performance may vary day to day. As degeneration advances, decline becomes consistent. Once structural loss crosses a density threshold, compensation collapses irreversibly. Cognitive reserve delays, but does not prevent this transition. Individuals with high reserve may maintain function longer, despite substantial pathology. However, once neuronal density falls below critical levels, decline accelerates regardless of reserve. Metabolic stress further limits reversibility. Chronic neuroinflammation, oxidative damage, and mitochondrial dysfunction create a hostile environment for recovery. Even surviving neurons function less efficiently in such conditions. Importantly, not all MCI progresses to dementia. Some cases reflect non-degenerative causes, such as depression, medication effects, or metabolic disturbances. In these instances, reversibility is possible because structural integrity remains intact. The distinction lies in underlying pathology. When MCI reflects neurodegenerative disease, the intervention window is defined by synaptic preservation. Once synapses and neurons are lost, therapy can only slow further decline. It cannot rebuild the original architecture. This reality underscores why early detection matters. Identifying MCI during synaptic dysfunction rather than after neuronal death increases the theoretical potential for stabilization. However, current therapies remain limited in their ability to halt progression completely. Irreversibility in pre-dementia network breakdown is anatomical. Synaptic loss becomes permanent when dendritic spines retract and cytoskeletal integrity collapses. Neuronal death is permanent. Axonal disconnection is permanent. The diagnosis of mild cognitive impairment may feel mild. The underlying structural changes may not be. The window between early dysfunction and irreversible collapse narrows as pathology advances. The fear embedded in this stage is biological constraint. Once the network's structural density falls below functional threshold, recovery is no longer possible. What remains is management rather than restoration. MCI is therefore a critical transitional phase. It may represent the last point at which structural integrity remains sufficient for potential stabilization. Beyond that, the architecture of memory and executive integration cannot be rebuilt. The pre-disease state, when the brain is already failing quietly. Mild cognitive impairment is often framed as a mild condition because independence remains intact, function is preserved, daily life continues. Yet biologically, MCI frequently represents a stage in which structural brain failure has already begun. By the time measurable decline appears on cognitive testing, synaptic density within hippocampal circuits has often decreased. Protein dysregulation may be active. Tau pathology may be spreading. White matter connectivity may already be thinning. The visible complaint of forgetfulness is not the origin of degeneration. It is the first detectable symptom of network breakdown. The brain does not fail suddenly. It destabilizes gradually. Synapses weaken before neurons die. Neurons die before large-scale atrophy becomes visible. Atrophy becomes visible before independence collapses. MCI sits in the interval between silent pathology and overt dementia. This interval is unstable. Compensation sustains performance temporarily. Cognitive reserve buffers decline. Frontal networks recruit additional resources. However, these mechanisms operate against ongoing structural erosion. The label mild refers to functional severity, not pathological intensity. The disease process driving MCI may already be extensive in microscopic and molecular levels. The absence of dramatic impairment does not imply absence of degeneration. Pre-dementia network breakdown is defined by loss of integration. Memory circuits lose synchronization. Executive systems struggle to coordinate. White matter disconnection slows signal transmission. These changes accumulate gradually until compensation fails. Independence is the final barrier separating MCI from dementia. When daily life becomes compromised, the diagnosis changes. The pathology does not. The biology has been progressing continuously. The fear within mild cognitive impairment lies in this hidden progression. The patient remains aware. Insight is preserved. They sense decline before others do. They function, but with increasing effort. They compensate, but with growing fatigue. Neurodegeneration, without overt disability, creates uncertainty. Not all MCI progresses. Some individuals remain stable. Others convert. The determining factor is underlying structural trajectory. Mild cognitive impairment therefore represents a pre-disease state in visible form. It is not normal aging. It is not simply forgetfulness. It is measurable evidence that neural networks supporting memory and executive integration are under structural strain. The progression from synaptic dysfunction to neuronal loss to network disconnection defines the pathway from MCI to dementia. Once structural thresholds are crossed, reversal does not occur. Understanding this mechanism reframes the diagnosis. MCI is not a reassurance of safety. It is a warning of vulnerability. It signals that the architecture of cognition is beginning to destabilize. The brain changes before memory loss becomes severe. It changes before independence collapses. It changes quietly, beneath performance thresholds, until compensation can no longer sustain function. Mild cognitive impairment is that moment of recognition. The moment when decline becomes measurable, but the network has already been weakening. This is not a metaphor. It is a structural sequence. Synapses weaken, connectivity thins. Neurons die. Integration collapses. The mind remains present while the infrastructure supporting it erodes. This is pathology of fear. And this fear is real.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.